And now the second speaker will be Diane Saunders from Vanderbilt University Medical Center. And she's going to talk to us about human pancreas development. Thank you, Diane.
Hi, everyone. My name is Diane Saunders, and I'm part of the Powers and Bursova Research Group at Vanderbilt University. Thank you for the meeting organizers for allowing me to share a small vignette of our work today. The pancreas is a bifunctional organ composed primarily of acinar cells, which secrete enzymes into a branched ductal network funneling into the duodenum. And scattered throughout the acinar cells are pancreatic islets of Langerhans, or collections of endocrine cells that secrete hormones necessary for regulating blood glucose. Despite their divergent functions, endocrine and exocrine cells share a common embryonic lineage, and both compartments, as shown in this schematic, are associated with vasculature, neuronal processes, and immune cells. As part of our efforts to build a human atlas of neonatal development and early life pancreas, or HANDLE-P, we collaborate with immunologists and hope that our pediatric tissue exploration and integration will provide insight into the development of autoimmunity and type 1 diabetes. Our team members coordinate organ procurement through a national network of nonprofit organizations, and we process tissue for imaging modalities in both traditional and multiplex. The cross-sectional data I'll show here is from donors divided into 3 stages, roughly based around developmental milestones and pancreas biology: neonatal, which encompasses the first 3 months of life; infancy, which spans 3 to 24 months; and childhood, which we consider ages 2 to 10. This graph and the 2 insets on the right show the rapid expansion of the pancreas by mass with increasing donor age. You can also appreciate that there is significant variability in organ size even at very early postnatal stages. I mentioned that the pancreas is a bifunctional organ, and this is easily visualized across tissue sections stained for hormones in green, red, and white. Just after birth, there are more small clusters of endocrine cells, particularly around the periphery of lobes, while in infancy and certainly by childhood, these cells have coalesced into larger islet structures with much more exocrine tissue in between them. We quantified these changes both by determining overall density of endocrine cells and by considering the relative proportion of different endocrine cell types. These data are captured in the graph on the left, which shows endocrine cell density decreasing from the neonatal stage to childhood, as well as an overall increase in the proportion of beta cells and corresponding decrease in delta cells. On the right, abundance of each endocrine cell type is plotted against pancreas weight, again with the highest percentages right around birth. In addition to shifts in cell density and composition, we also observe changes in islet architecture. Neonatal islets tend to have a beta cell dense core with alpha and delta cells around the periphery, which quite interestingly is reminiscent of a mature rodent islet. From neonatal to childhood stages, we've quantified enrichment for the intermingled structure that you see on the far right. We're also starting to correlate these changes with functional islet secretory profiles to better understand which signals might be driving the rearrangement. Our current work uses codex technology to simultaneously visualize 30 to 40 antigens on a single tissue section and thus more accurately define and classify cell phenotypes and states. This image is a neonatal pancreas where you can appreciate the pancreatic vasculature and islet capillaries, as well as immune cells and endocrine cells. We can also quantify mature acinar cells or identify mature acinar cells and an abundance of macrophages, which are the most prominent immune cell subtype. In addition, we detect all 5 hormone-producing endocrine cell types, including their rarer gamma and epsilon cells. And finally, we classify various immune lineages and record the spatial proximity of these resident or recruited cells to endocrine cells and vasculature. The final aspect of this project I want to mention is that we have made several hundred images available in an online interactive resource called Pancreatis, which you can find at pancreatis.org. This is intended to be a biologist-friendly platform, so the user interface is very simple, but it retrieves metadata and data from a backend database via API. Visitors can browse images from curated points of entry, which we call collections, filter based on characteristics of interest, and then launch a web viewer to further explore the images. The framework is available in GitHub if you have data you would like to make more accessible, or if you're interested in creating a navigational tool for your favorite organ or system. In conclusion, I hope I've helped you appreciate the plasticity of postnatal pancreas and the value of imaging modalities in constructing pediatric cell atlases. We have several projects investigating specific signals that are critical for islet cell development, like the transcription factors shown in this image, and plan to integrate with single cell sequencing data. This project has been a tremendous team effort, and I also want to recognize the extreme privilege and responsibility that comes from studying human pediatric tissue. I look forward to connecting with some of you at the upcoming poster session.
Perfect as well, right on time. Thank you. That's great.